So welcome everybody to this afternoon session. It's another session about polymicrobial interactions. So we heard already some talks about this, and it's great to have the opportunity to have some really great talks in this afternoon session. And I'm really excited that so many people came from this very nice plenary session. And yeah, just to introduce very briefly, how does this work? Yeah. So we heard already some talks about the co-infection in CF lung and about this polymicrobial environment. And today there will be some talks which really, figure out and pick out and look definitely into this interaction style of, colonization, infection, and it's not only the different bacteria and pathogens which impact the lung disease, but also the host response and the interaction with the host response. And I think this very nice figure from, it's already 2017 published, describes or alludes very nicely all the interactions which are going on between the pathogens and the epithelial, the host and the immune system of the host. And this only very brief introduction before I briefly introduce the speakers. So the first speaker will be Dr. Jonathan Cogan. He is at the Division of Pulmonary and Sleep Medicine in Seattle Children's Hospital. His special interest is about antibiotic treatment in CF, and his talk will be about treatment of co-infections. And the second talk will be given by Fabrice Jean-Pierre, a postdoctoral research fellow at the Geisel School of Medicine in Dartmouth. And his specialty is polymicrobial interaction, and his talk will be about modeling polymicrobial communities and antimicrobial resistance. The fourth talk will be given by Karen Moreau from Claude Bernardi University in Lyon, France. And her special interest is staph pathogenesis, and especially in this field, staph aureus Pseudomonas aeruginosa interaction. And her talk will be about staph aureus and Pseudomonas aeruginosa interaction. And the last talk will be given by Dr. Aurélie Crabet from the Laboratory of Pharmaceutical Microbiology in the University of Ghent in Belgium. And her special interest is host pathogen interaction in chronic lung disease, and her talk will be about immune modulation by resident flora, including anaerobes. And so I would like to ask John Cogan, and please step up. And I guess.
All right. Hi, everybody. Thanks so much for the introduction and as well as the opportunity to present. So as I was looking at the other speakers, I realized you guys are all amazing, I think, translational and basic science researchers, and I'm a lowly clinician who does clinical research. So this is going to be much more of an epidemiology kind of talk along with treatment of pulmonary exacerbations with respect to observational studies and any randomized data that's available. So just bear with me as we go. These are my disclosures. I am a part of two randomized controlled trials looking at pulmonary exacerbation treatment. Honored to be a co-investigator on those. So this is the outline for the presentation today. I'm a bit embarrassed after going to that amazing plenary and listening to Dr. West define pulmonary exacerbation, but you guys are going to have one more slide to make sure we're all on the same page related to that. Then we'll look a little bit at epidemiology and then some of the treatment guidelines before looking at polymicrobial infection prevalence during pulmonary exacerbation treatment. And then I was asked to look at the treatment approaches for co-infections in CF, of which there's not a lot of data, but we'll review the data that I was able to find, and then have a little brief word about antimicrobial stewardship in CF at the end. So as I mentioned, I suspect this is probably a review for most of us, but want to make sure we're all on the same page. So this table illustrates the common signs and symptoms consistent with the diagnosis of a pulmonary exacerbation. And these diagnoses can be made with subjective symptoms, including things like chest pain and fatigue, along with more objective findings like drops in lung function, weight loss, or increasing cough and speed and production. But ultimately, pulmonary exacerbation diagnosis is really made by treatment. And that generally includes the initiation of increased airway clearance along with the initiation of IV antibiotics, whether that's inhaled, oral, or IV. In terms of epidemiology, we saw these data already at the plenary, so I'll just be brief. But essentially, this figure comes from the CF Foundation and illustrates how common IV antibiotic treatments have been in children and adults living in the US. And so as you can see, for adolescents and adults, this number was fairly steady in the 30 to 50% range until about 2019, 2020, as Dr. West and Dr. Hoffman pointed out. And this was likely due, at least the decline is likely due to a combination of ETI dissemination as well as clinical care, along, of course, with the COVID pandemic. And then a similar trend was seen in children, though the baseline IV percentage was lower. And then the graphic at the bottom is taken just from the highlights of the 2022 registry, showing that the IV-treated pulmonary exacerbation frequency is still low, at least in the US, but is still occurring at least. So in terms of microbiology, here are just two figures taken from the Canadian CF registry and then the US CF Foundation patient registry. Again, this might be a review, I think, for most people in this room, but as you can see, the most common organism in both countries was Staph aureus, followed by Pseudomonas aeruginosa. And then the figure on the right is illustrating the Pseudomonas prevalence, and that has been declining over the past decade or so, again, likely due to improved eradication strategies, as well as overall improvement in the health of people with CF. Okay, so let's transition a little bit to how to treat a pulmonary exacerbation once they're diagnosed. So unfortunately, while pulmonary exacerbation guidelines and management recommendations exist, there are really limited data to support evidence-based strategies to inform antibiotic selection. And then with respect to co-infections specifically, there's really no mention on what to do when you have people with CF who have multiple organisms. So as you can see on this slide, the Canadian Pulmonary Exacerbation Guidelines, as well as a semi-recent UK antibiotic treatment document, suggests that respiratory culture positivity should guide antibiotic selection, but there's no specific comment on what to do when more than one organism is present. And then the 2009 US CF Foundation Pulmonary Exacerbation Guidelines, again, have no comment on what to do with that. So my editorial for you all is we really have no idea what to do. We don't know if we should be using antibiotics to cover all organisms present on culture. I'll try to walk you through a few studies that have begun to try to answer that question, but at the end of the day, there's really no hard and fast guideline that's currently in place. Okay, so first off, how prevalent are co-infections among people with CF? So this table is courtesy of Dr. Liz Cromwell. She's the director of biostatistics from the US CF Foundation. It was nice enough to put this together for me. It's a busy table, but it's meant to show you the number of people with CF who have cultured 1, 2, or 3 or more microorganisms by year. So you can see on the far left column is the year, and then the various number of microorganisms that were present in each of those years. So to make it less busy, I really just wanted to highlight the years 2019 and 2020. And as you can see in 2019, close to 50% of people with CF cultured 2 or more microorganisms. But then in 2020, this number dropped to around 30%. And again, the reasons for this are likely multifactorial, but probably include the initiation of ETI into clinical practice, and then also a reduction in the number of cultures collected. And then as Dr. Hoffman mentioned about an hour ago, that we're getting OP swabs instead of sputum, which perhaps are less sensitive in picking up some of these organisms. What is equally interesting, though, I will say, in my opinion, is comparing the years 2020 and 2021. So as you can see, the drop in the frequency of 2 or more microorganisms between 2019 and 2020 appears to also be sustained in 2021. And this might be due, again, to the same limitations that we mentioned, or this might be really a real trend. So keep these numbers in mind, and this is really nothing to do with pulmonary exacerbations in terms of the timing. It's just related to the overall prevalence among people with CF in the US CF registry. So I wanted to look at studies that looked at the prevalence of co-infection prior to pulmonary exacerbation treatment. So these data come from Dr. D.B. Sanders and the original STOP study that Dr. West mentioned in the plenary. I feel like all I'm doing is recapitulating the plenary. I apologize to you guys out there. Anyway, so as you can see, the vast majority of people in the study, and this is in adults 18 and older, had cultures that were positive for Pseudomonas. You can see there it's about 71% with just Pseudomonas, and then 56% with the mucoid version. But there's no mention in this study, at least in the tables or anywhere in the results, about the number of people who cultured more than one respiratory microorganism. So what about children? So this, I think, is a really excellent study looking at the prevalence of co-infection prior to pulmonary exacerbation treatment. And this comes from Anna Fano, and Anna used the CF Foundation Patient Registry Pediatric Health Information System linked dataset, or the CFFPR-FIS linked dataset. And the CF registry provides really excellent outpatient information related to demographics, clinical information, along with microbiology, while the phys data set includes all the antibiotics that are utilized. So this study included about 5,000 children who contributed close to 28,000 pulmonary exacerbation. And among these, 73% were classified as polymicrobial, or having more than one of the typical respiratory organisms present. So to orient you to this figure, this is an intersection plot. On the y-axis is the number of pulmonary exacerbation. On the x-axis are the various organisms and the frequency in which a polymicrobial infection is present. So as you can see, as an example, MRSA and Pseudomonas were present together in close to 3,000 pulmonary exacerbation. That's the highest of the bars. And then the one next to it is MSSA and Pseudomonas, and they were present together in more than 2,700 pulmonary exacerbation. Ms. Fano then looked at the proportion of pulmonary exacerbation with what she termed full antibiotic coverage. So full antibiotic coverage was defined if antibiotics were prescribed that had activity against all the respiratory organisms that were present. So the y-axis here describes the proportion of pulmonary exacerbation with full antibiotic coverage, while the x-axis is the number of hospitals that contributed data to the study. So as you can see, among all polymicrobial infections, the median pulmonary exacerbation full antibiotic coverage was 68%, with some variation within each hospital. So about 2/3 of the time when you have more than one organism present, people have been using antibiotics to cover all those organisms. That makes sense. In terms of the specific combinations, this is looking at the 5 most common combinations. And so again, to go back to that tallest bar from the last figure, there were over 2,800 pulmonary exacerbations in which Pseudomonas and MRSA were present. And when they occurred together, there was 82% of the time antibiotics were used to treat both of those organisms. And you can see a slight decline as you go down. So another example would be the last row on this table. If you were colonized or infected with Pseudomonas, MSSA, and then Stenotrophomonas, you were treated 57% of the time with antibiotics to cover all of those organisms. Okay, so switching gears a little bit before we get into the pulmonary exacerbation treatment strategies, I wanted to share results of a little bit of an older study. The objective of this one was to assess the effects of Pseudomonas and Staph aureus infection on lower airway inflammation in children. And these investigators looked at bronchoalveolar lavage fluid from 111 kids with CF, and more specifically, examined inflammatory markers and other markers and indicators of disease severity. This is the most basic science translation I get, so pay attention, I guess, to those people who like this. On the first column, you'll have the log of the total white blood cells, and you can see some of the other parameters and markers that they studied, and then what those numbers were in the presence of various organisms, whether you had none cultured, 1, 2, or 3. And then just to kind of highlight, I suppose, the most relevant results. If you squint, you can see that the total number of white blood cells increases with the number of pathogens present. I think a little bit more interestingly is the amount of neutrophil elastase, which is a marker of inflammation that has been associated with worse clinical outcomes, appears to increase with more microorganisms present. So these results provide at least some data suggesting that perhaps the presence of one microorganism might be associated with worse clinical outcomes. I suspect you're going to learn a lot more of that from the other speakers in the next hour and a half or so. Okay, we'll finally get to what I was asked to do, which is to look at whether treatment of some or all co-infections is associated with improved clinical outcomes. I'm going to show you guys results from 4 studies, which will hopefully provide some guidance moving forward. Of note, as you will see, the term co-infection is admittedly used a bit loosely for many of these, but hopefully you will get an idea of what I'm trying to illustrate. Okay, so this first study was published more than 20 years ago, and the authors aimed to compare pulmonary exacerbations with single and multiple organisms in people with CF with chronic Burkholderia cepacia infections. And the authors hypothesized that having BCC plus another bacterial isolate would lead to a larger inflammatory response compared to infection with just BCC. So a really small study. They looked at 13 adults with CF who had chronic Burkholderia cepacia infection, and they selected the antimicrobials based on in vitro susceptibility of the co-infecting organisms. So people with organisms other than BCC essentially were given antibiotics to target those organisms. So as you can see from this table, when comparing the outcomes between people with CF who were infected with BCC alone, and that's the single isolate portion of this table, versus BCC and another organism, there were no statistically significant differences in any of the measured outcomes, including clinical outcomes like lung function and BMI, and then more of the inflammatory markers like CRP. So the authors concluded that pulmonary exacerbations with multiple organisms are no more severe than those where only BCC was isolated, and that perhaps treatment of those extra co-infections, if you will, was not associated with any improved outcomes. So the next study comes from the CF Matters group. This was a randomized control trial that compared standard antibiotic therapy to standard therapy plus an additional antibiotic based on the microbiome sputum analysis. And the idea here was to determine if microbiome-guided antibiotic therapy would be associated with improved outcomes. So the forest plot illustrated here compared the control arm to the active arm at discrete time intervals. And at each interval, there were no significant differences in lung function recovery by arm. So these results would suggest that microbiome-guided antibiotic selection is not associated with improved outcomes. So in this example, I used microbiome as a co-infection. So it's a little bit, not orthodox, but it's an attempt at trying to answer the question. I will say I ran into Dr. Plant yesterday, very nice guy, you guys know him, who told me that they're in the process of submitting the definitive manuscript, so hopefully it'll be out for publication shortly. Okay, the third study to briefly discuss comes from the group at University of Michigan, Dr. Kastner and colleagues, and these investigators aim to compare outcomes of pulmonary exacerbations treated with broader anaerobic coverage versus minimal anaerobic coverage, with the thought that perhaps treating anaerobes might be associated with improved outcomes. So you can see here in this table that broad anaerobic coverage was used in 20% of all pulmonary exacerbations. And this table is their table 1, showing differences in lung function and microbiology between the two groups. As you can see, people treated with broader anaerobic coverage had a lower baseline lung function and also higher colonization with the Chromobacter xylus oxidans. So here are their results. And as you can see, treatment with broader antibiotic coverage was not associated with a larger change in lung function pre to post pulmonary exacerbation or a reduced hazard of a future pulmonary exacerbation requiring IV antibiotics. So these results seem to indicate that there might be opportunities to be more targeted with respect to antibiotic selection in the treatment of a pulmonary exacerbation. In this case, I used anaerobic coverage as a way to kind of look at co-infection, so like anaerobes essentially. All right, but we're finally going to get to one that perhaps answers the question, maybe the reason why I'm here. I was fortunate enough to work on a study for this population. So this is again, to go back to Ms. Fano's study, this is looking at co-infection with MRSA and Pseudomonas. So we were interested in looking among these children whether treatment with both an anti-MRSA antibiotic and an anti-Pseudomonal antibiotic would be associated with improved outcomes in this group compared to treatment with just an anti-Pseudomonal antibiotic. So in this case, the co-infection is MRSA with Pseudomonas. And again, the purpose of the study was really to try to get a sense of whether multiple organisms needed to be treated or if antibiotics targeting one would be sufficient. So this is a bar graph illustrating the proportion of pulmonary exacerbation treated with both anti-MRSA and anti-Pseudomonal antibiotics by the participating hospital in the study. This was using that CFFPR Phys dataset that I mentioned earlier. As you can see, there's really substantial variability among centers in terms of treatment. And then also some centers always cover both organisms with antibiotics while others do it much less. And there really is variability among centers, but also within centers, which is kind of interesting. So what we found was that the addition of an anti-MRSA antibiotic to standard anti-Pseudomonal antibiotic therapy was not associated with improvements in any of the measured outcomes when compared to treatment with an anti-Pseudomonal antibiotic alone. We specifically looked at changes in pre to post pulmonary exacerbation lung function, which is not shown on this table. And then the odds of returning to lung function baseline or the odds of having a subsequent pulmonary exacerbation requiring IV antibiotics, which is present on this table. So these results suggest that children with CF co-infected with MRSA and Pseudomonas may not benefit from the addition of anti-MRSA antibiotics for pulmonary exacerbation treatment. The last couple of minutes, I just wanted to briefly have a quick discussion about the role of antimicrobial stewardship in CF. The primary goal is really to optimize clinical outcomes while minimizing any unintended consequences of antibiotic use. And this includes defining the need for antibiotic treatment along with optimizing the choice, dose, and treatment duration for the antibiotics given. And I would suggest that while the data that I just mentioned are all really observational in nature, they do seem to provide some evidence suggesting that limiting antibiotic exposure in the treatment of co-infections for a pulmonary exacerbation is possible. So for anyone interested in this topic a little bit more, I would encourage you to take a look at this review from Dr. Lloyd, published in the Journal of Pediatric Infectious Diseases last year. And this review provides practical approaches that CF care teams can take to incorporate antimicrobial stewardship principles into CF care. So for example, requiring pre-approval for various broader spectrum antibiotics can help to provide more targeted antibiotic therapy, reduce antibiotic-related side effects, and then avoid redundant antibiotics. And in addition, the use of local pulmonary exacerbation guidelines and clinical pathways can help to generate new data that can guide clinical practice, including more studies that help to determine the potential benefits or perhaps detriments of treating co-infections. I just want to lastly finish up with a couple examples of some of these guidelines. So this is a study from Kennedy et al., and actually Dr. Hahn is one of the authors on this study. And this was an attempt at trying to create an empiric antimicrobial algorithm for treatment of pulmonary exacerbation. It's a pretty busy slide, so I would just encourage you to look at the publication if you're interested in learning more. But essentially, they walk through recommendations on what to do based on the respiratory organisms that are present. And I think it's a really nice way to try to get at standardizing treatment and getting at some of these antimicrobial stewardship issues. And then we similarly did something, this is from Seattle Children's. We worked with our stewardship team on this particular guideline. And so, for example, in the lower left-hand corner, you can see recommendations for people in the presence of MRSA and Pseudomonas. Now, I will say this guideline was created in 2019, and my hope is to go back to the group and maybe consider adjusting this guideline based on that study that I just showed you. And perhaps a consideration can be given to just start by treating Pseudomonas alone and then adding on MRSA therapy if needed later during the hospital stay. So in conclusion, polymicrobial infections are certainly common when people with CF, perhaps going down in the setting of ETI or perhaps not, maybe a little too early to really say. Evidence from observational studies seems to suggest that antibiotic treatment targeting all CF-related microorganisms may not be necessary. And ultimately, prospective studies are needed to determine optimal antibiotic selection for pulmonary exacerbation among people with CF who culture multiple CF-related microorganisms. I think this could certainly be a question. You know, we learned about the James Lind Alliance priorities, the plenary, and I think this is one that could be teed up in the future either for the STOP group or for some other investigators to consider looking at. I just wanted to briefly acknowledge a number of people, including the workshop chairs, again, for the invitation to present, some of my mentors and funding, and happy to take any questions. Thank you.
All right, so we invite anybody who has questions to come to the microphone, please.
Thank you very much. I'm John Bradley. I'm a pediatric infectious disease doctor. It's wonderful to be at my first CF meeting, and I'm very impressed. We don't have the kind of plenary session focus with our patients that you do. It's very inspiring. But I do spend a lot of time in the microbiology lab, and I think every patient with CF has co-infections. And it's not just one or two bacteria, it's the normal flora that gets in there. And you all know this, you've been living this, and it's extremely complicated. The microbiology lab actually is designed in their protocols to pick out certain organisms. So you can have a plate full of organisms and just a few Pseudomonas, and what they'll report is Pseudomonas with normal flora. And I encourage everyone to go down to the microbiology lab and actually look at those CF plates because they're impressive. There's no other infection that gives you, except maybe ruptured appendicitis, it gives you so many bacteria on one plate, and you're treating them all. And so some of the insight that if you use an antibiotic that's not active against the organism you isolate, and they still get better, I think it's because you're treating all those other things. So I'm adding to the complexity and hoping to work with you all to try and figure this out at some level. Thank you.
Yeah, thanks so much for your comment. I think it's very well said. At the end of the day, we really don't know what we're doing with our antibiotics in CF. And I could show you dozens of studies that have looked at just the ridiculous amount of variation. At the end of the day, people in general kind of get better. It almost kind of doesn't even matter what antibiotic you pick, in my opinion. And I think that's a good example with some of the studies that we walked through, that maybe you just start with the narrower approach, narrower. You know, for us CF doctors, narrow is still perhaps more broad for the ID doctors. But if you start a little bit more narrow, you may get the same outcomes.
So I had a question for you as well. It's a little off topic, but what about viral co-infection? How do you think that plays a role in selecting antibiotics?
Oh, man. This is definitely not my wheelhouse. There's probably 2/3 of the people in this room, including yourself, who could answer this one better. I guess what I would say, I'll go back to my wheelhouse and then say the Stop Peds study is perhaps a good example of what to do. So this was, again, in the plenary. Here I am again, once talking about the plenary. So the Stop Peds pilot was a study that looked at whether immediate antibiotics for outpatient pulmonary exacerbation in children is associated with basically better outcomes than just starting with increasing airway clearance alone. And what they found was you could avoid antibiotics 2/3 of the time. And I'm going to suspect, based on other studies, that the vast majority of the time, outpatient exacerbations in children are virally triggered. So I would think based on that, and there's lots more work to do to really look at it, but there might be opportunities to avoid antibiotic exposure entirely in the presence of viral infections. I know that's one of the questions that we're going to look at in the larger, more definitive Stop Peds trial. I don't know if you have any other thoughts on that either.
Good answer.
Yes.
All right. Well, thank you so much. So I'd like to invite our next speaker, Dr. Jean-Pierre.
That's pretty great. It's really bright up here. Okay, I guess I'll have to stick with that. All right, so good afternoon, everyone. Thanks to the organizers for inviting me for this really amazing session. I'm really excited to be here. So today, I'm Fabrice. I'm almost no longer a postdoc at Dartmouth, soon to be assistant professor in January. I'm really excited to be talking about a project I've been working the last couple of years that really seeks to understand how microbes in the cystic fibrosis airway can interact among them. So I've got no disclosures. So this crowd already knows that a hallmark of CF lung disease is the accumulation of a thick mucus layer in the airway of these individuals. Gotta have a pointer? Okay, it doesn't work. And that mucus that is rich in nutrients constitutes of an ideal environment where different microbes can colonize and cause chronic infections. And these infections, unfortunately, are hard to treat with the utilization of antimicrobials that are currently available in the clinic. And this is a really interesting observation because if some of you remember your basic microbiology classes, most of the approaches that we use to study microbial pathogenesis has been inspired by this work that has been done by Dr. Robert Koch, that scientist, German scientist from the 19th century, which pretty much said that a microorganism must be isolated from a diseased organism and grown in pure culture. And this is really interesting because if you go back into the literature that has been published by Dr. Anderson in the 1930s, if you delve into the paper, you can clearly see that she has used such approach. Whereas you can see here, she mentioned that a pure culture of Staphylococcus aureus was obtained from the lung, or plural, from one person that was suffering of cystic fibrosis. But one of the problems that arises by using such approaches is that we know that microbes in the cystic fibrosis airway are hard to treat. And here I'm just showing you a list that I've gotten from the CF website where we look at the antimicrobials that are currently used to treat chronic airway infections. We have a bunch of them. So we have penicillins, cephalosporins, aminoglycosides, vancomycins, etc. And all of these compounds can pretty much show a really nice antimicrobial activity against microbes in even in vitro settings. So here I'm showing Staphylococcus aureus, Pseudomonas aeruginosa. They can be pretty much targeted against by these antimicrobials. But when we're looking back in the clinic, we see that it does not work. And one of the hypotheses that can explain why these microbes are really hard to treat is because they have the capacity to interact among them. And indeed, in the last couple of years, there are some mechanisms that have been defined to really be critical in shifting the susceptibility of microbes, specifically in the context of a polymicrobial community. And these mechanisms can include the production of enzymatic inactivation of molecules such as beta-lactamases, where a member in the community can produce a beta-lactamase that will confer passive protection to an otherwise sensitive member in the community. You can also have the transfer of genetic materials such as plasmids that can confer antimicrobial resistance to other members in the community, but you can also have metabolites that can be produced and change the physiology of other microbes that are grown specifically in a polymicrobial environment. And to really bring it home and convince you folks, I hope I don't have to convince you, that interactions matter in the context of chronic infections, I just want to give you two examples. So first, on the right-hand side of the slide, so what we're looking at is the lung function of people with CF as represented by FEV1. So basically, the higher the bar is, the better the lung function is. So in that work that was published by Nicola Moley a couple of years ago, she has seen that people with CF that are both colonized by Staphylococcus aureus and Pseudomonas aeruginosa had worsened lung function compared to individuals that were only colonized by either Pseudomonas or Staph or neither of these two microbial species. In the second example that I'm showing you on the left-hand side of the slide, where at this time we're looking at the impact of vancomycin, that is a frontline CF drug, targeted specifically against Staphylococcus aureus biofilm cells, as represented on the y-axis, we see that indeed, following Koch's postulates, vancomycin is capable of eradicating a couple of logs of biofilm cells of Staphylococcus aureus. But when you're taking metabolites or molecules that are produced by Pseudomonas aeruginosa and treating those Staphylococcus biofilm cells in the presence of vancomycin, well, we pretty much see that vancomycin is not really capable of eradicating Staphylococcus aureus. So interactions matter in the context of airway infections. But now one of the problems that arises is that, okay, well, we know that Staphylococcus aureus and Pseudomonas aeruginosa can interact, but which model should we be working in the lab? And this is a pretty complicated question, because if we're looking at the microbiome of airway microbiome of people with CF, so basically using next generation sequencing, looking at 16S of microbes that are detected in sputum samples, what I'm showing you here is a bar that represents an individual with cystic fibrosis, the different colors representing the different microbial taxa that you can see on the right-hand side of the slide. So we can see that this individual is colonized by some Prevotella, by some Streptococcus, by some Haemophilus. But the more that we sequence different samples from different people with CF, the more that we see that the composition is really heterogeneous, which really makes it not easy to find one specific community type that we could model in the lab and study at the molecular level. And coming up with models, in vitro models, to understand those interactions and how they can exacerbate CF lung disease is a really critical question that we need to tackle. And we were interested in tackling that question at Dartmouth. So instead of saying that it would be possible to identify one specific community that is responsible of most of the negative outcomes in people with CF, we hypothesized that it was perhaps a limited number of community compositions could encompass most of the microbial diversity observed in the cystic fibrosis airway. And to really get to that question, work that has been pioneered by Dr. Tom Hampton, a bioinformatician at Dartmouth, where he looked at studies, including hundreds of patient samples from people with CF, he managed to identify that there's at least 5 different community types that can encompass most of the microbial diversity observed in the airways of people with cystic fibrosis. So we can see that there's a community type that is dominated by Streptococcus species, another one, cluster number 2, dominated by Pseudomonas aeruginosa, a third cluster that is more of a mixed bag of lowly prevalent pathogens, but when they're there, they're really abundant, and 2 other community types, P, M1 and M2, that are basically, when we're using other computational approaches, we have seen that it's pretty much one similar community type. So for today, because at the time, I'm only going to be talking about that same cluster, then I'm going to be saying that it's a mixed community. So now we have bioinformatics predictions, but now if we want to go back to the lab, the first question that we had to ask is like, okay, well, which microbes should we be picking now and model in vitro to study those interactions? And we've decided to focus on those 4 different microbial species that are Pseudomonas aeruginosa, Staphylococcus aureus, Streptococcus species, and Prevotella for multiple reasons that I'm going to try to summarize in only one slide. So first of all, we know from microbiome studies that these microbes are among the most abundant and prevalent in the cystic fibrosis airway. We have also used metabolic modeling, which is an in silico predictive tool that allows you to reconstruct the metabolic potential of any living organism. And we have seen that predicted metabolic interactions between those 4 microbes could explain most of the metabolic interactions detected in the cystic fibrosis lung. I've talked earlier about the interactions between staph and Pseudomonas that can lead to worsened airway outcomes in those individuals. There's also some work that has been published by Dr. Diane Newman's group where she used a tissue cleaning method, MyPack, and then she has shown that some of these microbes can colocalize together directly in sputum. And finally, but not the least, we also have some evidence showing that interactions with at least 2 of these microbes can have an impact on their susceptibility to antimicrobials. So now, how to build a biofilm, or a polymicrobial biofilm, I should say. So we picked those 4 microbes that we decided to grow in CF-like conditions. So using artificial sputum medium, we decided to grow them as either monocultures or in a mixed community using a 96-well plate. And when we grew them for 24 hours, a community or preformed biofilm forms at the bottom of the plate. We take out the non-attached cells and we feed the biofilm with fresh medium for an additional 24 hours. And after that total of 48 hours, from the time 0 to time 48, we can quantify the outputs as CFUs per mil. And I always like to show that picture because we get lots of colors, so it's really exciting, and we can generate a lot of data. So the first piece of data that I want to present to you today is here. What we're going to be looking at is the number of biofilm cells as represented on the y-axis by lactin CFUs per mil. When the bar is lightly colored, it represents the microbe grown by itself. When it's darker, it's the microbe grown in a mixed community. Not surprisingly, Pseudomonas aeruginosa doesn't really care whether or not it's in the presence of someone or not. For Staphylococcus aureus, and this also echoes what other studies have reported, Staphylococcus grows a little bit less in a mixed community versus monoculture. Streptococcus is pretty much, I consider it like someone that always needs a friend, so that when there's someone, oh yeah, super happy, grows pretty well. That's what we've seen in our system. And lastly, but not the least, Prevotella, that is an anaerobe in our system, basically we could not get it to grow by itself in artificial sputum medium, but only in a mixed microbial community. And I hope that you have some time to go and see my colleague's poster, because Bassam El-Hafi, a graduate student in our lab, has used molecular approaches to really understand how those interactions are driving that phenotype, and has identified that short-chain fatty acids are really important in driving the capacity of Prevotella to grow, specifically in a polymicrobial environment. So this was really nice, but we also wanted to take things a little bit further. So we also tested clinical isolates of Pseudomonas aeruginosa, MRSA strains of Staphylococcus aureus, the Streptococcus miller group that is also important for exacerbation in vets and people with CF, and also a couple of Prevotella species. So now, if we take a step back, loop back to the beginning of my talk, I've been talking about antimicrobial susceptibility. Now that we had a model that we could work with, the question was to really understand how those interactions can shift antimicrobial susceptibility of these pathogens specifically grown in a polymicrobial environment. And today I'm going to be talking about tobramycin. Why? Because tobramycin is one of the most prescribed antimicrobials to treat airway infections in people with cystic fibrosis. And I'm also going to be only talking about Pseudomonas aeruginosa, but there's a lot of things that are happening in that system. So here we're going to be looking at the number of cells of Pseudomonas aeruginosa represented on the y-axis. The P represents planktonic cells and the B represents biofilm cells. So we can see that when we're taking clinically relevant concentrations of tobramycin, not surprisingly, we completely eradicate planktonic cells of Pseudomonas aeruginosa. But when we look at biofilm cells, we see that there's tolerance of these biofilm cells of Pseudomonas aeruginosa, which is pretty much already known. But when we were looking in a mixed microbial environment, we were really surprised because we saw that while there was tolerance of Pseudomonas aeruginosa biofilm cells monoculture in the presence of tobramycin, while Pseudomonas was again completely eradicated in a mixed microbial environment. And we were like a little bit puzzled because this was not really reflecting what is seen in the clinical reports. And to really get to that question, we decided to use an approach where we're looking at mutants of Pseudomonas aeruginosa that are frequently detected to emerge or detected in the context of airway infections. And I'm not going to go into the mechanisms of LASAR, but what we know is that, first of all, it's prevalent in the cystic fibrosis airway. We also know there's some work showing that LASAR mutants can also drive exaggerated pulmonary inflammation responses. The LASAR mutant, some beautiful work done by Dr. Deb Hogan's lab and Deb Hogan's group at Dartmouth shows that LASAR variants have growth advantages in low oxygen conditions, but also that LASAR variants can correlate to negative outcomes in people with cystic fibrosis. So we had a couple of arguments to really see and test what is happening with a LASAR mutant in our system, and that's what we did. So looking at pure culture of Pseudomonas aeruginosa wild-type mutant and also complemented strains, in the presence of tobramycin, they pretty much all behave the same. But in a mixed microbial environment, when we were doing the same experiment, we saw that LASAR mutant was pretty much not showing that increased susceptibility to tobramycin versus monoculture versus the wild type. And for molecular biologists, this is the holy grail because it allows you to ask the question, what's the mechanism? Because of the time, I'm not going to go into all the specific details, but one of the targets of the LASAR system are phenicines. So for those of you who do not know what phenicines are, I'm showing you the structure of the molecule here. So it's under the oxidized state here, but it has a capacity of shuttling electrons, so gaining or losing electrons. And one of the best ways to see the impact of phenicines in Pseudomonas aeruginosa is looking at the color of an overnight culture. So when the culture has been well shaken, we have that really blue-greenish pigment that is produced because the phenicine is oxidized. But if you take that tube and let it sit on your bench for a couple of minutes, it goes back to its reduced state and then it turns yellow. And there's some beautiful work by the Dietrich and Newman groups that have shown that there's an impact of phenicines on the capacity of Pseudomonas aeruginosa to tolerate different types of antimicrobials. So we decided to test that in the lab, and we decided to go back in our system, and we built the mutants. And then when we treated those different strains with tobramycin, again, in the context of pure cultures, we didn't see any differences in how they behaved, either the wild type or the mutant strains. But when we looked in the polymicrobial environment, we were really happy on that day because by blocking the production of phenicines in the LASAR background, we're basically allowing tobramycin to eradicate Pseudomonas aeruginosa. And again, that brought us the question to know, well, is it because there are more phenicines that are produced in the LASAR mutant in a mixed microbial community? And we decided to test that hypothesis. And indeed, by using HPLC coupled to mass spectrometry, what I'm showing you here on the left is that those are pure culture strains of these different strains. We see that all the controls pretty much behave how they're supposed to. But in a mixed microbial environment, our hypotheses are pretty much validated. So basically, Lassa mutants in a polymicrobial environment produce more phenocins than the wild-type strain. And again, the other question that we had was to ask the following: if it's possible to induce tolerance of Pseudomonas aeruginosa by exogenously adding phenocin back to our system, and then we had tested the hypothesis. So basically, here we remember wild-type Pseudomonas aeruginosa in a mixed microbial environment is further sensitized to tobamycin versus monoculture. And by adding back phenocins into our system, we saw that we were capable of inducing the tolerance profile of Pseudomonas aeruginosa in the presence of that antibiotic. Again, the loaded question: how do phenocins promote tobamycin tolerance of Pseudomonas aeruginosa in a polymicrobial environment? If we go back to the impact of phenocins on the physiology of Pseudomonas aeruginosa, well, there's several things that phenocins can do to Pseudomonas aeruginosa. It can act on biofilm matrix production, on efflux pump, but also on metabolism. And I don't have the time to show you all the data, but we've pretty much ruled out, at least in some part, the impact of biofilm matrix and efflux pumps in driving that phenotype that are driven by phenocins. But we have definitely seen something interesting as it pertains to the metabolism. And when we think about metabolism, we can also think about electron shuttling. So Pseudomonas aeruginosa has 5 terminal oxidase systems, at least that are known, that have the capacity to act in different conditions to allow it to thrive in different conditions. And there's also some nice work showing that phenocins can interact but with the CCO1 and CCO2 terminal oxidases that are the high affinity oxygen one. And so we had the hypothesis that we wanted to test that was basically, well, do those terminal oxidases contribute to the tobamycin recalcitrance of Pseudomonas aeruginosa, specifically in a polymicrobial environment? So we tested the hypothesis. So this is new data that we had just generated. Again, you remember Lassa mutants in the mixed microbial environment, they're tolerant to tobamycin, whereas the other strains are all susceptible. When we're adding in phenocins without tobamycin, they also all behave pretty much all the same. But this was really exciting to us because when we're adding back phenocin to either the wild type, the Lassa or Fez or double Fez Lassa mutant, we see that we can trigger tolerance, but in any conditions where we abolish the activity of those 2 terminal oxidases, we can never induce a tolerance of Pseudomonas aeruginosa, which really suggests that these terminal oxidases are really important in driving tolerance of Pseudomonas aeruginosa to tobamycin. So to wrap things up, we built a model that is of clinical relevance to CF airway that we use to study how those interactions can have an impact on drug susceptibility. We were kind of surprised to see that by adding in tobamycin, that Pseudomonas was disappearing from the system. So we decided to use an approach where we're using mutants of Pseudomonas aeruginosa that are detected in the cystic fibrosis airway. And one such mutant codes for the Lassa gene, for the Lassa protein. And then we saw that Lassa variants could thrive again in a mixed microbial environment in the presence of tobamycin. But looking at the molecular level, we saw that phenocins and also terminal oxidases seem to be really important in driving that tolerance phenotype of Pseudomonas aeruginosa in a polymicrobial environment. And with that, I would like to thank the lab, also the collaborators, the fundings. And again, I'm going to be opening my lab soon, so happy to chat more. If you want to collaborate, please just reach out.
So again, any questions, if you guys can come to the microphone. So I had a question for you about the polymicrobial community that you originally created. So I think for the Streptococcus, there's a lot of understanding of the commensal nature of the Streptococcus, but Prevotella, I think there's some literature suggests that some are maybe more commensals and some are more virulent.
Yeah.
So how did you kind of decide which species to include in the model?
So we've decided there's some nice work published by Dr. Surratt's group where he's basically looked at all the different types of Prevotellas that are detected in the cystic fibrosis airway, and the specific species that is the most prevalent is Prevotella melanogenica. So it's pretty much a dominating Prevotella species in a cystic fibrosis lung. There are also other species such as intermedia that are common, but we decided to go with melanogenica given that it's really prevalent in a cystic fibrosis airway.
Hi, Fabrice, Jade Andikar from the University of Washington. That was really beautiful work. And it might have been a few years, but since somebody mentioned LASAR at CF. So Dallas Mold and Deb Hogan have this really nice story about how LASR mutants and the wild type can cross-feed each other and sort of heighten pathogenesis. And I was wondering if you've taken a mix of LASR mutants and the wild type, put both of them into your mixed species biofilm, and if you got differential susceptibility to tobramycin.
I did that, but that experiment was a little bit complicated because the LASR mutant has already a growth advantage over the wild type. So you're kind of like confounding several factors at the same time. But this is definitely something that we want to optimize and see if we will be able to like still drive the tolerance of wild type Pseudomonas in the presence of LASR. Yeah, that's a cool idea.
All right. Thank you so much. I'd like to invite Dr. Barreau up as our next speaker.
Thank you, everyone. And I want first to thank the organizer to give me the opportunity to present what we are doing in our lab. And so I will talk about molecular interaction between Staphylococcus aureus and Pseudomonas aeruginosa. It is not very original, I will say. And I don't know how to move and okay, and I have to say I have nothing to disclose for this presentation. So as you all know now, there is two major bacteria that colonize the lungs of people with cystic fibrosis, and the first one is Staphylococcus aureus, which is the one who colonize a young patient, and we can have up to 80% of young patients that are colonized by this pathogens. But the percentage of patients that are colonized by Staphylococcus will decrease with the age. On the contrary, we will observe an increase of percentage of people colonized by Pseudomonas aeruginosa according to the age of the people. In this context, we can have up to 40% of people, CF people, that are co-colonized or co-infected by these two pathogens. And what we're wondering is what's going on between these two bacterial species and why they are so often presently together in the lung. When we started the project, there had been two interaction states that had been described for these two bacteria. The first one, I don't know if you see, no, you don't see, maybe. Not easy. The first one is competition. When Pseudomonas is able to inhibit the growth of Staphylococcus aureus. As you can see here on in vitro test, if you seed an agar plate with Staphylococcus aureus and put a spot of Pseudomonas in the middle, you will see an inhibition halo around the Pseudomonas aeruginosa. You also see in gross culture, when you had Pseudomonas, you will observe a decrease of Staphylococcus aureus growth. But there was another interaction state that have been described that we call it coexistence, in which both bacteria, both species can grow together without affecting the growth of each other. When we started, the type of interaction is defined by Pseudomonas aeruginosa, which can be either competitive or coexistence with Staphylococcus aureus. When we started this project, there was only few papers that described few strains of Pseudomonas aeruginosa that were in coexistence. Okay, I've lost it. Okay, and so we're wondering what happened. It's not easy to do. We're wondering what happened for a clinical strain isolate from people with cystic fibrosis. To answer this question, okay, I don't know. Yes, to answer this question, we isolate 52 couple of strains, pseudo and staph couple of strain, isolate for co-infected patient. And we did this in vitro test. And in fact, we show that in 65% of the case, they were in coexistence, meaning that if we study a clinical strain, we have most of the strains that are in coexistence. And it is really different what we observe with our laboratory strains. So if coexistence is so prevalent, we're wondering what happened in this context, what is the impact of coexistence on these two bacteria. To explore this question, we did a transcriptomic analysis using RNA-seq, and we've done some in vitro culture of Pseudomonas aeruginosa and Staphylococcus aureus that we compare to co-culture. On first point, we looked on the impact of Pseudomonas aeruginosa on Staphylococcus aureus transcriptome. And here you have a representation of the number of genes that are dysregulated, upregulated in black and downregulated in gray, in the context of coexistence or in context of competition. What you can observe is that we have more genes that are dysregulated on coexistence. And today we will focus on transporter genes. And more specifically, we looked for, I will talk about the Nord genes family that includes the TET38 genes of Staphylococcus aureus. And what we observe is that in context of Pseudomonas aeruginosa, we have an increased expression of these genes. And it's specific to interaction of Pseudomonas aeruginosa, as we did not observe this overexpression with other species. And we also demonstrate that we need a close contact between the two species to have this overexpression. Why these categories, this family of genes was interesting is because we knew that they encode for efflux pump and they are involved in antibiotic resistance. So we're wondering what could be the impact of Pseudomonas aeruginosa in antibiotic sensitivity of Staphylococcus aureus. To explore this, we made some monoculture of staph or co-cultures that we submit to two types of antibiotic, tetracycline and ciprofloxacin. And we looked at the survival rate of Staphylococcus in co-culture versus monoculture. In the graph, when you have a rate of 1, it means that there is no difference of survival rates between co-culture and monoculture. That is the case in the absence of antibiotic. But if we expose our culture with antibiotic, we observe a higher rate, meaning that in the presence of Pseudomonas aeruginosa, we have a better survival rate of Staphylococcus aureus. The TET38 genes was also described to be involved in internalization of staph. So we explore the impact of Pseudomonas aeruginosa in this phenotype. To do this, we infect epithelial cells with monoculture of staph or co-culture with Pseudomonas aeruginosa during 2 hours, and after we made some gentamicin treatment to eliminate extracellular bacteria, and after we collect the intracellular and determine the number of intracellular Staphylococcus aureus. As you can see, if we infect with monoculture of staph, we have about 5% of Staphylococcus that will be internalized in epithelial cells. But in presence of Pseudomonas aeruginosa, we have a higher rate, about 15% of staph that will be internalized. And it is a very important phenotype because we know that the internalized Staphylococcus aureus is a way of the bacteria to escape from immune systems. On the second part, we look on the other side, what is the impact of Staphylococcus aureus on Pseudomonas aeruginosa transcriptome. And here you have another representation, but that show you also the number of genes that are dysregulated in Pseudomonas aeruginosa in presence of staph, and according to the categories of genes. And what we observe that we have more genes. Again, we have more genes that are dysregulated in coexistence in comparison to competition. And more specifically, we observe that in coexistence, we have a huge modification of gene expression involved in carbon and amino acid metabolism. So we looked more precisely in this way, and what we observe, we have downregulation of genes involved in glycolysis and the use of glycose, but a high overexpression of genes that are involved in amino acid catabolism, but also, and more interestingly, in genes involved in acetoin catabolism. Why acetoin was interesting? It's because it has been described in the literature that Staphylococcus aureus is able to produce acetoin. And here we see that in presence of staph, Pseudomonas increase this metabolism. So we confirm this data on our clinical strain, and we show that in yellow, staph is able to produce acetoin during growth, and in red, we confirm that Pseudomonas is not able to produce acetoin. And in co-culture in purple, you have an intermediate level of acetoin, meaning that maybe Pseudomonas is able to use the acetoin produced by staph. To confirm this hypothesis, we cultivate Pseudomonas aeruginosa in a staph supernatant containing acetoin. And as you can see during the growth, we have a decrease of acetoin in the, relating to the growth of Pseudomonas aeruginosa. So we confirm that Pseudomonas is able to use the acetoin produced by staph. But why is it using this acetoin? To look for this question, we use a Pseudomonas aeruginosa wild-type strain in blue and 2 mutants that are not able to use acetoin anymore in gray. As you can see, if we cultivate this strain in a minimal medium, in which we put only acetoin as carbon source, we show that Pseudomonas wild-type strain is able to grow, but strain that are not able to use acetoin can't grow. So Pseudomonas is able to use acetoin as a sole carbon source for its fitness. After that, we did a long-term co-culture, 5-day co-culture of Pseudomonas and Staphylococcus aureus using the same Pseudomonas aeruginosa strains. And you see, if we use a wild-type strain that is able to use acetoin, we have a better survival rate in co-culture than in monoculture, meaning that in the presence of staph, Pseudo grow better in co-culture. But if we use Pseudomonas strains that are not able to use acetoin, we don't see the better survival. That means that using acetoin produced by Staphylococcus aureus, Pseudomonas aeruginosa has a better fitness in a long-term co-culture. Okay. To conclude on this first part, what we have demonstrated is that when we study clinical strain of Pseudomonas and Staphylococcus aureus, in 65% of the case, they are in coexistence. And in this context of coexistence, what we observe is that Pseudomonas aeruginosa induce an increase of antibiotic resistance of Staphylococcus aureus and a higher rate of internalization, which allows staph to escape from immune system. On the other side, Staphylococcus aureus will produce acetoin, which allows Pseudomonas aeruginosa to have a better fitness to use this molecule as a carbon source and increase its survival rate in the context of nutritive competition. So more than coexistence, we think that bacteria cooperate in this context. So, okay, we found that some of our clinical strains are in competition and others are in coexistence. So the next question we ask is it possible there is a switch between the two interaction states? To explore this question, we first made an in vitro evolution experiment. For that, we use on your left we use a competitive strain, and on your right, coexistent strains that we cultivate on the top without Staphylococcus aureus and on the bottom with Staphylococcus aureus. And we made 30 subculture to induce the evolution of the strains on plate. And every 5 subculture, we looked for the interaction state with Staphylococcus aureus. At the end of this experiment, if we look of the competitive original isolates, we made 20 replicates of the experiment. And among the 20 replicates, you see that 11 of the isolates evolve towards coexistence, and 9 stay in competition with Staphylococcus aureus. So we have an evolution from competition to coexistence. But if we look on coexistent original isolates, after the 30 subculture, we don't see any evolution. Coexistence strain stay coexistence with Staphylococcus aureus. So it seems that we can have an evolution from competition to coexistence. But what about what happened in vivo? Is there the same evolution from acute infection, first infection in patient, and chronic infection in patient? To explore this question, we use a collection of clinical strains from a unique patient that we followed during 6 years. We started in 2011, which is the first date that the patient was colonized by Pseudomonas aeruginosa. And since 2011, he was chronically infected with Pseudomonas aeruginosa and with 2 strains of Staphylococcus aureus. So we collected all the strains during these 6 years. And first we made some competitive assay, but we did it in a broth culture to be allowed to define a competition index. And as you can see here, the first strain of the patient is highly competitive against Staphylococcus aureus, and the last strains of the patient are coexistent with Staphylococcus aureus. In the middle, we have a very variable index of competition index. But what we can observe is an evolution from competition to coexistence. After that, we made a whole genome sequencing of all the Pseudomonas aeruginosa strains, which allow us to demonstrate that it was only one unique strain that evolved over the time. And what we see, it's an evolution from competition in red to coexistence in green, according to the time. And we added on this phylogenetic time tree the mucoid phenotype, so you can also see that the strain evolve towards mucoidy isolates. We also demonstrate that we have mutation in mutel or mutes genes of Pseudomonas aeruginosa, meaning that there are some hypermutator isolates that explain the evolution of the strains. We wonder if there is a link between this evolution toward coexistence with other phenotype of Pseudomonas aeruginosa. So we looked for different phenotypes. The first is growth, and we determine the double time of each isolate we have. And as you can see, according to the time and the evolution of the strain, the doubling time increase. And if we compare the coexisting strain isolates to competitive one, we can see they have a higher doubling time. We also looked for a biofilm capacity of biofilm formation for all the isolates, and you see it's very variable according to the isolates. But if we compare coexisting strain isolates, they are less capable of doing biofilms than competitive one. And we also looked for virulence factor expression by RT-qPCR, and we looked for major regulator of virulence depending of the current sensing of Pseudomonas aeruginosa, as you already heard last year, but also PQSA, RHLR, and MVFR. And we look for genes involved in the biofilm and mucoidy phenotype. And we have shown that coexistent strains are less virulent as all the 4 major regulators we looked are are less expressed in coexistent strains in comparison to competitive one. And we also observe that we have a higher expression of LGD genes, which is involved in alginate formation that can explain the mucoid phenotype, but we did not observe any difference in mucoid genes. This data will be confirmed by RNA-seq analysis to have a global vision of dysregulation during the evolution. So in conclusion, what we have shown in this second part is that we have in vivo an evolution from competition to coexistent strains. And in these coexisting strains of PA, they have different phenotypes. They grow slower, they are mucoid, they make less biofilm, and they are less virulent. And what we have previously shown is that in coexistence, we have a bacterial cooperation, and it may be factors that favor persistence of infection. So to finish, I want to thank my collaborator, and especially these 4 young, brilliant researchers that made the work, and also my collaborators and our funding foundation, and especially the French Cystic Fibrosis Foundation. And I want to thank you for your attention. And thank you.
So I had a question while we're waiting for people to come up to the microphone. The clinical data, so you said that the reason that the bacteria evolved from competition to coexisting is because it increased their growth and their sustainability, but the virulence factors go down. So for the patient that you followed that over time, did you notice like decreased exacerbations or anything that would suggest that having 2 less virulent organisms might be better than having one that's more?
We try to make a correlation between the outcomes, clinical outcomes, and the type of interaction between staph and pseudo. And honestly, we didn't see anything very very convincing. Yeah. But the problem is we worked only on our collection in our hospital, and I think the collection was too small to have a real effect. But I don't know if there is in my point of view, maybe there is no clinical difference, but we have to consider if staph is a helper of Pseudomonas to persist in the lung of the patient. And if so, it may be a factor that favor chronic infection. But indeed, the presence of staph, I'm not sure, and I agree with what was said before about co-infection. I'm not sure, and what we see in the literature, the presence of staph, it doesn't seem to be major, I don't know what to say, it doesn't increase exacerbation of worse clinical outcomes. But we have to consider maybe it favor the implantation of other bugs and maybe have other implications.
That was a great talk. Thank you, Karen. My question is similar to Andrea's. With your original collection of the coexistent and the competitive, did you notice that they had more, that the coexisting or the competitive had more in children versus adults? Did you have the data to look to see whether you had more pediatric cases, had the competitive, and as you had the coexistent was more in the adult?
Curiously, we didn't see any difference in age of the patient regarding the 2 types of interaction. And we have young patients that have already coexisting strains. But to be honest with you, when we made this first study, we only picked 1 isolate per patient. And I think it's not the best way to look at. After we made a small study in which we took about 10 isolates from different patients in the same sample, in the same sputum of the patient. And we look for some patients, all are in coexistence, all are in competitive, but for the majority of the samples, we have a mix of competitive and coexistence isolates. So from my point of view, maybe in the lungs of the patient, we have the 2 phenotypes, only maybe during a period of co-infection, and maybe after we have only one phenotype. And it will be very interesting to look of this evolution in one patient, but taking different isolate at each point in time to really have a real image of what happened in the lung.
Thank you.
So further questions? I don't see. So Karen, very nice. Thank you. So did you ever see that Staph aureus has a deleterious impact on Pseudomonas?
Never.
Never.
We tried.
Yeah, but so in some of our patients, we also can see that Staph aureus overgrowth is able then, is more than Pseudomonas and can replace Pseudomonas aeruginosa from the lungs of CF patients. But you never saw something like this?
But I will be very interesting in looking at these strains. No, but we know there are some, if we calculate competitive index, we see for one Pseudomonas aeruginosa strain, it won't be the same with different Staphylococcus aureus. Mm-hmm. So it must be a cross discussion between the two, but we don't know exactly how it works.
Mm-hmm. Okay. Pavel.
Thank you. So very interesting work. So I wonder if you had the chance to check the quantity of both bacteria during the course of infection. So if there is something like for competition that there would be like more of Staphylococcus versus Pseudomonas, and then going to some steady state in, like, if you look at the microbiome data, for instance, to see that the abundance would be similar during the phase of their coexistence.
We have to look more further like this in a huge amount of patients, I think, and samples. But what is strange is in some samples, clinical samples, that they have determined that we have higher rates, higher level of staph and than Pseudomonas aeruginosa, even if in our in vitro tests they are in competition. So I don't know if there is a real... It's a very complex question of what we are looking, what we have in our sputum and what we looked in our...
This is what I wonder. It might be the competition because Pseudomonas needs to have Staphylococcus under the control. So
yeah, but I don't know. I'm not a physician, but I always wonder what we collect when we have the sputum, which part of the infection we have. And I suspect that in the lung, we can have different niche of infection, and some are in competition for what we are studying. Some we can have competition between staph and pseudo, and in some niche we can have coexistence that is not explored at all, the diversity of phenotype and interaction in different areas. So I don't know if we can correlate what we looked. And as I told you, if I be honest, what's the mistake we've made is to collect only one isolate per samples. And if we answer your question, we have to look more precisely of the diversity of interaction in the sample.
Okay, thank you very much. So now I would like to ask the last speaker to come to the podium, and Dr. Krabi will talk again about the co-infection.
No. I did something wrong? Yes. Okay, go ahead.
I don't know.
Oh, gosh. Sorry. I did something wrong. I'm so sorry.
No, I don't know. So is there any help around?
I got outside.
No, my computer.
Do you have a USB with you?
No.
You can plug in and play if you have.
Do you have USB? So I don't begin. There was somebody sitting there. Yeah, no,
On my computer.
no, it's open. Maybe home. Yeah, okay. And no,
Yes,
and then
okay.
yeah. Okay. Start 5?
Yes.
I'll start. Should start. Yeah.
Okay, sorry for that. I just pressed the wrong
Collaboration.
button. Okay, that's a great start. So thank you very much for having me today. And I'm very excited to be talking about how microbes from the lung microbiome could actually influence inflammation. I'm also going to be talking about anaerobes today. Okay. Okay, so the lungs of patients with cystic fibrosis are characterized by bacterial dysbiosis. If you would compare to a healthy lung, you would have a typical transient community. We're not very sure what their role is really in immunity and all of that. However, it's very transient. In patients with chronic lung diseases like cystic fibrosis, there is more a persistent microbial community because of the altered microenvironment, the sticky mucus. In a healthy lung, you would count about 100 CFU per mil of lung fluids. However, in people with cystic fibrosis, the CFUs can reach up to 10 to the 10th CFU per mil of sputum. So there is a big difference between a healthy lung and lungs of people with cystic fibrosis. The main issue there is, of course, the bacterial burden in the lungs, but also the dysbiotic community. So overall, people with cystic fibrosis have a higher abundance of typical pathogens like Pseudomonas aeruginosa or Staphylococcus aureus. We all know those pathogens very well. We've been studying them for so many years, and we do know that their presence and their abundance is related to poor clinical outcomes in patients. However, in addition to these classical pathogenic species, there are also many different microorganisms present. As for example, here from the group of John Puma in the past here, following up the microbial composition over time in a patient with cystic fibrosis, you do see lots of green and yellow for Pseudomonas and Staph. However, you also see many other colors, so many other microorganisms being present. These are, among others, facultative anaerobes like Rothia, Streptococcus species, but also many anaerobic species are present in the lung microbiome. However, their role in the disease process is until now quite unclear, and there's a lot of work going on in that regard to really understand what their role is. How could these non-pathogenic, or let's say the species which are typically considered as non-pathogenic in people with cystic fibrosis, how could they influence CF lung disease? Well, they could have a direct impact on pathogenesis, for example. So they could influence the growth of Pseudomonas or other pathogens. They could indirectly influence pathogenesis by interacting with the virulence factor production of these pathogens. They could influence immunity, the immune response, or they could interfere with the activity of antimicrobials, as also Fabrice has been describing earlier. For today's talk, I'm going to be focusing on the immunomodulation of these non-pathogenic species. How I'm going to approach this? So I'm going to first walk you through a selection, because I couldn't describe everything today, but I'm going to select a few clinical association studies in which researchers have associated microbial composition and the abundance of certain species with specific clinical outcomes like lung function or inflammation. Then I'm going to be talking about in vitro work and in vivo data on immunomodulation of these resident flora. I'm going to be discussing interactions between these clinical association studies, because some of them have actually been a starting point for further in vitro and in vivo validation, and also to more understand the functional properties of these microorganisms, but also vice versa. Some in vitro and in vivo data has also been confirmed in patients to really further understand the clinical relevance of these interactions. And then ultimately, what's of real interest to everyone, of course, is the clinical relevance of these findings, and could this lead to new applications to treat people with cystic fibrosis and also perhaps other chronic lung diseases. So let's start with the clinical association studies. Early work here from Edith Zemanik's group has actually, as I mentioned before, so tried to correlate the abundance of certain microbial species to clinical outcomes or certain parameters which are important in the disease process. Specifically here, they looked at lung function, expressed as FEV1, and the levels of different pro-inflammatory cytokines. Let's start with the most logical example. If we would look, for example, at Pseudomonas and Staphylococcus abundance, you would see their abundance positively correlated with the levels of certain pro-inflammatory cytokines. That does make a lot of sense, right? But at the time, it was, for me at least also, and I think many of us, surprising to find an inverse correlation between the abundance of certain of these other typical non-pathogenic species and inflammation. Very specifically here, Rossia, for example, was inversely related to the levels of pro-inflammatory mediators, Prevotella as well, and other species as well. More recent work has really confirmed some of these findings, and there are many studies out there. So apologies if I'm not citing yours today, but there are many out there. So again, finding positive associations between typical pathogens like Pseudomonas and negative health outcomes, and other species like here Rossia again being linked with more positive health outcomes. Let's also just look a little bit beyond the CF field, because I think we can also learn from this insight, or at least confirm some of these findings. Some very impactful work recently was published in which species of Prevotella were positively associated with different health outcomes, such as the COPD severity, for example, or the exacerbations, so less exacerbations with higher abundance of these anaerobes like Prevotella. Also here, a study from James Chalmers' group showed a positive association between Rossia and then less severe disease, less exacerbations, etc. So just a few examples here. We have also analyzed these kind of data sets. So we were focusing on 3 study groups at the time, not people with cystic fibrosis, but still I feel relevant for the audience. Bronchiectasis and also severe asthma, also characterized by inflammatory phenotypes. And for these 3 groups, we had matching 16S data as well as inflammatory mediators. And we collaborated here with Grant Rogers and Stephen Taylor at SUMRI in Australia, and with James Chalmers at University of Dundee. So our aim was really to kind of use these matching data sets, but slightly differently as had been done before. So we wanted to look at the extremes, right? We wanted to look at the patients that had very, very high inflammatory levels based on these markers, and the other extreme, those that had very, very low inflammation. We wanted to understand which species or genera would discriminate between these 2 extremes. So we looked at the 25th and the 75th percentile. So that's a question we wanted to answer, right? So which taxa would discriminate both groups? We did that using LEFC analysis. Without going into details, it's an algorithm that exactly allows us to answer this question. And just to give you one example here, Rossia, you get like an LDA score, so it really associates the abundance of a specific species with a specific group. In this case, Rossia was positively associated here in green with low inflammation based on interleukin-1 beta, and what Pseudomonas, for example, had the opposite. It was associated with high inflammation group, which is not surprising. So these were the results actually from the 3 different cohorts and the 8 different markers that we had included there, really summarizing the data. And you can see that many of the different taxa are associated with low inflammation, and a couple are associated with high inflammation, like Pseudomonas. I'd like to draw your attention to our top species, which were most, or taxa in this case, which were most associated with low inflammation group. So again, we're recognizing Rossia, we're recognizing Prevotella. I won't be talking much about Neisseria because in CF it hasn't been studied that much. And then maybe for those of you who were paying very close attention to this graph, I left one out. And not because I don't want to show it to you, but just I want to build up the suspense, and I'm going to come back to this a little bit later in my talk. So don't leave the room right now, because you might want to hear. Okay, so those were the clinical association studies. Now let's look at in vitro and in vivo data on immune modulation. What we have done to answer this question, we typically use like physiologically relevant 3D cell culture models of long epithelial cells using the rotating wall vessel bioreactor. I can tell you more about that later if you're interested. But basically, these models are differentiating into in vivo-like structures, right? So they express tight junctions, for example, mucus. So that's our model. Then we have our pro-inflammatory stimulus relevant for CF. We use Pseudomonas aeruginosa, LPS, or virulence factors. And then we have our collection of CF sputum isolates, which we obtained using protocols developed by the Surrett lab. So trying to isolate as many species as we could from CF sputum samples. So that's a collection that we obtained from several sputum samples. And then we tested that. We brought the long epithelial cells together with the pro-inflammatory stimulus and the bacteria, and saw if they would influence inflammation or not. This is a summary of a lot of data, and a little bit has shifted here, so apologies for that. What we're looking at here, so we're looking at our inflammatory outcome, the activation of NF-kappa-B pathway. It's a very important pro-inflammatory pathway, important in CF and other lung diseases. We tested 2 doses of our candidates, anti-inflammatory bacteria, and expressed as MOI, multiplicity of infection, so the number of bacteria you add per epithelial cell. Let me just pick out one example here. So we have Actinomyces nice-lundi. This is expressed as percent inactivation of NF-kappa-B pathway, and NF-kappa-B pathway was activated with LPS. So LPS by itself is 100%. You add your bacterium, all the way it goes to, in this case, and it's very tiny, 3%. So the conclusion here is that we found different anti-inflammatory species to be part of the lung microbiome, but their activity can really depend. You know, sometimes the pathway gets inactivated stronger than for others. But also, if you have a look at Prevotella, within the same genus, different species would differentially inactivate the NF-kappa-B pathway. So we have differences there as well. We explore this further. I was hoping the screen would be even bigger to be able to show the data more closely, so apologies if it's a bit tiny. But we aim to understand if there would be differences in the minimal effective dose that you would need to inhibit inflammation between, in this case, our 9 anti-inflammatory species that we discovered. This is again expressed as MOI, so multiplicity of infection. I'm going to focus on, let's pick one, Prevotella. I think it's Stolica here, yes. And so with an increasing dose, you get a higher inactivation of NF-kappa-B, but you would need at least a dose here of, what was it? This is very tiny even for me, so sorry. I think you need a dose of 3.3 for those that can read better than me, so apologies if that's not the right number, but you need a certain dose, right? And so that dose depended depending on the species. What we found is that out of these 9, we had actually our top 3 of the most potent anti-inflammatory species. And there we gave a little gold star to Rosia mucilaginosa, because for that bacterium, at least in vitro in our 3D model, we would only need 0.2 bacteria per epithelial cell to decrease inflammation. While other species, we would need like 12.5 or even 50 to inhibit inflammation. Talking more about Rosia. So Rosia, we had actually found that Rosia has very potent anti-inflammatory activity in vitro, first of all. Here we're looking again at 3D cells of CF epithelial cells, bronchial or alveolar epithelial cells. And so Rosia was able to inhibit the production of different pro-inflammatory cytokines when stimulated with Pseudomonas aeruginosa, LPS, or any other kind of pro-inflammatory mediator we would add to the culture. And also this effect was really not strain specific. So we tested a lot of different Pseudomonas clinical isolates. We tested lots of Rosia, mucilaginosa, and other species of Rosia isolates, including CF isolates. We even tested isolates from healthy individuals from other lung diseases. Our student Charlotte, who was performing this project, even swapped her cats and got a Rotia from the cat. And so all of them were actually having the same anti-inflammatory effects. What about in vivo? So we used an in vivo mouse model to see if that would also be applicable in vivo. We performed intratracheal instillation of LPS to generate inflammation, expressed as MIP2, or the human homologue of IL-8. And there as well, LPS triggers MIP2 production. Addition of Rotia decreases that again. Looking at histology, we also found that LPS was triggering inflammation and lung damage, but addition of Rotia was, at least the CF isolate of Rotia, was again bringing lung histology more towards a more normal histological perspective or phenotype. What about patients? We all know mice are not patients, so we also wanted to see if our findings would be relevant for patients, or if we could find clues from these data. And so here we collaborated again with Geraint Rogers at Stanmore in Australia. And so we looked at sputum samples of 100 patients with bronchiectasis. And so we quantified the levels of Rotia mucilaginose using qPCR and also the levels of pro-inflammatory cytokines. And we found consistently an inverse correlation. So the more Rotia you would have in the sample, the less inflammation you would have. So somehow this, of course, it's still an association study, but it is somehow giving us confidence that our findings that we obtained in vitro and in vivo are hopefully relevant for patients. All right, to just also show some data for these other bacteria. These are publications from other groups and then ours. There have been other reports on Prevotella. So I showed before Prevotella was anti-inflammatory in our models as well. Other groups have shown this before. There are some contradictory findings in literature. I think some researchers see Rotia more as potentially pathogenic species that contribute to the infection process or to the disease process, while others could see it as potentially beneficial. Here in one study, this was with Prevotella intermedia. Their neutrophilic infiltration in the lungs of animals was reported after introduction of supernatant of Prevotella in the lungs, similar to Pseudomonas. So there are some contradictory findings out there, which I would also like to highlight today. Finally, also Streptococcus has been shown to be anti-inflammatory in different studies. This is here a recent study, very elegant, where they also screened many different isolates from CF sputum. And many of them were also anti-inflammatory, but they focused on Streptococcus because they were so strongly anti-inflammatory. And these effects were confirmed in precision cut lung slices of animals, again, seeing the same anti-inflammatory effects. To come back to this. So as I mentioned, we had one species here, which we had not found to be previously associated with low inflammation. And you might not have heard of this because it's not very abundant or frequently found in CF patients, but it might be interesting from a therapeutic point of view, maybe one day. It's called Aeruginosa. Have you heard of it?
Yes.
Okay. Maybe I'm the only one who hadn't heard of it, then maybe that's possible. So we actually found it also to be very frequently associated with a low inflammation group. And so we performed some additional analyses, this time correlating the whole data set. So the levels of Aeruginosa with inflammatory mediators, and we found for many different, for different cohorts, not all of them, but different cohorts and different markers, an inverse correlation between the levels of Aeruginosa and inflammation. This gave us clues to see whether that would or wouldn't actually be meaningful in vitro. And so we tested the effect of Aeruginosa species in vitro as well, again, using our 3D model systems. Long story short, using different triggers, LPS or Pseudomonas aeruginosa, we were able to see that 2 species of Aeruginosa, and I'm not sure who invents the names of these species, but I haven't practiced enough to be able to pronounce this, so I'll just call it AC and AP. And so they were actually, both of them, somehow able to inhibit inflammation. Sometimes it depended on the stimulus. You you would need a higher dose, but overall we would see an anti-inflammatory effect of this species or this genus. Okay, final part. What is the clinical relevance and are there potential applications of all these findings for people with CF? Well, taking together the current evidence on immune modulation, both based on clinical association studies and in vitro and in vivo analyses, of course, considering that there are some controversies in literature, I think it is, I can be quite confident to say, but I think we need more research, of course, that facultative anaerobes like Rothia species and non-pathogenic Streptococcus species, that they might be anti-inflammatory. And strict anaerobes, in particular Prevotella species, might also have anti-inflammatory properties. This might all depend, of course, on the context, on disease severity, patient-specific aspects, the microenvironment, many aspects to consider, but I'm talking about the current evidence so far in the context of CF. So then the question comes up, should we treat these microorganisms or should we purposefully not treat them? That's a question which I hope we will all be able to find an answer to in the future. I think we need to collect more evidence to make sure that they are safe, these species to keep, before we think of strategies to maybe use them from a therapeutic point of view. We all know inflammation is a double-edged sword. We all need inflammation to fight pathogens, but in patients with chronic lung disease, we have too much inflammation, so dampening that would be nice. But of course, we also need to look at other aspects like effects on antibiotic susceptibility and all of that before we can decide on that. Finally, these insights could form a base for microbiome-based interventions. We could think of targeted antibiotic therapy. Can we think the unthinkable and perhaps think of supplementation of these bacteria in the lung microbiome? We're far from there, but it's something maybe to think about if we're open to that option. And finally, this could lead to new insights for the identification of postbiotic compounds, so beneficial microbial compounds, which could also be used potentially for therapeutic interventions. With that, I would like to thank mainly two PhD students, Ellen and Charlotte, whose work I have presented today, the whole team back in Ghent. Merel is here as well. She's presenting a poster, and I would like to thank many collaborators involved in these studies, including our clinical partners, Geraint Rogers, James Chalmers, and also many others involved in this work. And thank you very much for listening.
Okay, please, Paul.
I'm Paul Planet from UPenn and CHOP. I did my PhD on aggregated bacteria Actinomycetium comitans.
we should talk. Yeah.
So the reason, one of the really key distinguishing features behind AA is that it metabolizes short chain fatty acids, and I wonder if its ability to metabolize short-chain fatty acids actually maybe decreases, is the reason why you're getting an immunomodulatory effect.
Yeah, indeed. Like it's been shown that short-chain fatty acids can be present in the lung environments of people with CF and other lung diseases. However, in contrast to, but indeed, because in the gut it's shown to be anti-inflammatory, right, but in the lungs there's more evidence that it could be pro-inflammatory. So that could be an interesting theory.
It's also quite pro-inflammatory where AA causes disease, which is in the periodontal pocket.
Yeah, okay. Right. Very interesting, thank you. We should talk further later, so okay.
Hi, Jeroen Verhuizen from UCSD. Great talk. I just had a question. When you guys were collecting the sputum sample, was that collected in multiple occasions? We know that CF lungs are heterogeneous. We know that they're also affected temporally. Different times they will produce different organisms, especially on the broad-range PCR. So my question to you, was it obtained in different time periods, and also when they had exacerbation? And if so, did you guys notice any difference with these anaerobes?
That would be very interesting to study. So these are single time points sputum samples, and actually these studies were already available, so we couldn't add more at the time, of course, but it could be interesting towards the future to look at that as well. There are differences in lung microbiome composition. We know it as a function of time, so that could definitely influence that. So yeah, let's look at that in the future for sure. And your second point was?
Yeah, that was my point.
Oh, was it? Oh, okay, all right, easy.
All right, thank you so much.
Thanks for a lovely talk. I'm Valerie from Sick Kids in Toronto. A question about your association that you showed between IL-8 and Rothia density. Do you have data on the number of antibiotic courses that these patients had? Because that could be a very important confounder. Patients who have higher antibiotic courses will have higher inflammation in their lungs because they're sicker and may have less Rothia as well because of a more restricted microbiome. So I think that would be an important confounder to look at, just in case you get a review.
No, absolutely. And so actually these patients were recruited as part of the BLESS study. And so these were actually in which they had they were testing the effect of azithromycin on these patients, and those were the ones that had not been exposed to azithromycin, so the control group. So, but I'm not sure if they had other antibiotics, so it would be indeed good to have a look at that. Thanks for your comments.
Hi, great talk. Thank you very much. I'm Ida Samanik from University of Colorado. I think one of the challenges that in some of the CF data has been, does less inflammation mean no inflammation? And is it anti-inflammatory if you're seeing less inflammation? Because I think in our studies and in some of the work, like BAL work from the REST CF data, they've seen that you get increased inflammation in the presence of mixed anaerobes, but it's not as much as with Pseudomonas. So in comparison, it looks good, but it's probably not a positive for our patients. It'd be better if they had nothing. But do you have thoughts about that?
I agree, and that's indeed like the double-edged sword, right? We do want to dampen the overt pathological inflammatory response, but we still want to have an innate immunity in the patients. So I think it's really going to be about understanding these interactions further, and then perhaps we could, if we think about these kind of therapies, we could select species which are not completely dampening inflammation. Maybe a combination of species could be an approach, because we've also showed some species are having synergistic interactions. So we also have to consider that, because if you already have anti-inflammatory species in your lung microbiome, and let's say some kind of approach, you would increase that, while synergistic interactions can bring it even further down, which is something to consider, because we want to make sure that we don't dampen it too much, of course. So absolutely.
Hi, I'm Paula Zamora from Dartmouth. In your in vitro experiments where you were using the A549 cells, and you see that with Pseudomonas you have like the common increase in IL-8, and then you add Rothia and you see that goes down with that, just like those two bugs. Do you know the mechanism, or have any idea of the mechanism by which like that goes down?
Yes. So I hadn't had a chance to discuss that. So thank you for giving me the opportunity to actually show that. And actually, I have a slide ready. Perfect. Yes. So thank you so much for that question. Yes, so we actually found that Rothia does not, in our experiments at least, because these are often short-term experiments, it's not like having any anti-microbial properties on the pathogens, because it's also inhibiting LPS-induced inflammation, for example. So all of our experiments point out towards a host inhibitory and host-targeted anti-inflammatory effect. So it seems to be inhibiting the NF-kappa-B pathways. So we did that using qPCR analysis. We found different genes to be downregulated when we add Pseudomonas versus Pseudomonas plus Rothia. And so we confirmed that using, among others, and there's much more data, using an NF-kappa-B reporter cell line. So that seems to be the mechanism, like really host-directed.
Very related question, Dan Wozniak, Ohio State. So if in your mouse experiment with this, you added LPS and Rothia together at the same time, I'm just wondering, if you start inflammation and then come in later with Rothia, do you still see an anti-inflammatory?
Yes, so we haven't done these experiments in vivo. However, we have done that in vitro. And so when you would first add your pro-inflammatory stimulus for like 24 hours and then you add Rothia, it still inhibits it. So it still works this way as well.
Thank you.
Thank you.
Okay, so I have a question as a clinical microbiologist. So usually in the cultures of our patients, we often see Rothia, and we don't know if you should report or just, And so I guess from your data, we really should report Rothia, because this might be, what do you think?
That's a tricky question. I would say that there is more research needed, of course. I think all the data so far points towards a potentially beneficial role, but then the question is, what would you do if you would report it? And you say, okay, Rotia is there. The clinician would have to decide, oh, Rotia is there, so we would have to think about choosing an antibiotic which is not killing Rotia. We have done these kind of tests to see if there would be antibiotics working against Pseudomonas and not against Rotia. They do exist. But yeah, it's really more research is needed. We have to test that to make sure that could be helpful for patients, and then reporting and indeed having a decision based on that would be helpful. Yeah.
Okay. And, sorry, maybe a last question about the Staph aureus and the anti-inflammatory effects. So, do you have any data on Staph aureus?
Yes, actually. Oh, I don't have an extra slide for that. I'm sorry. So yes. So in our initial work, we were actually co-culturing Pseudomonas together with the different species, including Staphylococcus aureus. We could not find an anti-inflammatory effect or a pro-inflammatory effect of the combination. I know some researchers, I think Dao and Guyen as well, has published an anti-inflammatory effect of Staphylococcus, if I'm not mistaken, but we haven't found that in our experiments. So it can also be, again, very strain-dependent environment, media-dependent. Yeah.
Okay,
absolutely.
thank you.
Great, thank you so much.